Hi everyone, I'm Nick Alexander. Today I'm going to be telling you all about, well, what I've learned about the pre-processing of these data in SPM. And, what do I mean by pre-processing? Well, I suppose it's simply the signal processing steps which occur after you've collected your data and before you do your analysis. So I suppose that's it. And what's involved in pre-processing? Well, you should have some data set. Maybe you downloaded it, maybe you're planning it. But yeah, you should have some data set which will be a bunch of files, including some kind of raw neural time series data, as well as some sort of peripheral information about, I don't know, event timings, perhaps a table of timings or an extra channel in your data. You might, for EEG, have electrode labels to tell you where those channels were recorded from. For MEG, you may also have positions and orientations, information about grad structures, that kind of thing. And you may also have knowledge about the anatomy you've been recording from and where that is in relation to those channels. Anyway, the most important thing that you'll have is some knowledge about what it is you've actually recorded. And it might seem kind of obvious, but that's going to guide all of your pre-processing as well as your analysis. Hopefully you know a little bit about what the signals you've actually, some of the properties of the signals you've attempted to measure. And you should hopefully also know something about the noise environment in which you were kind of forced to record those signals along with. And yeah, like I say, that's going to guide everything you do. And there's no one-size-fits-all kind of standard pre-processing pipeline in EEG and MEG. Although I guess there are enough commonalities across recordings that, yeah, there are a few sort of standard principles which I guess I'll raise as we go along. But I just want to say, you're going to be the person who knows your data best, and yeah, just kind of go with what you think's best and what other people have done in the past. Anyway, the idea is that at the end of it, you're going to end up with an isolated signal, and that's going to allow you to run your analysis sort of across your design, and you'll hear about how to do that in some of the later talks. And yeah, why do it? What am I even going to be talking about? Some people have suggested perhaps that certainly in the case of EEG, good luck with MEG, but certainly in the case of EEG, it could be better to kind of leave the data alone. And this is essentially a paper which presents quite a clean data set with a nice simple experimental design, and shows that if you just apply, I think it's a high pass filter, and then just use averaging across lots of trials, you can get the same result that you'd get as if you kind of, I'm not sure, I think they manually went through and also applied some sort of semi-automated methods to remove artifacts, to select bad channels, bad segments, all that kind of stuff, and showed that, yeah, it doesn't help. So yeah, why do it if that's enough? And yeah, it's enough if your data's clean and you've got lots of trials and a simple design, but you'll usually find that artifacts in your data are not a small problem that you need to get around. You'll find that they're orders of magnitude larger than any of the signals in your data normally. So these might be physiological, so that would be signals arising from other parts in the body. It's not just the brain that produces kind of electrophysiological signals. Unfortunately, persistence, there's the heart, so ECG or EKG, and there's also muscle activity. So muscles from the neck in particular are a problem in EEG data sets. And they might also be nothing to do with what you were trying to record or anything from the subject. They could be non-physiological. They could be reflecting some real other magnetic or electrical source in the world. So light noise, for example, that kind of 50 Hertz AC source, which we tend to pick up with EEG and MEG, unfortunately. 50 Hertz in the UK. or perhaps it's something which is not real at all. It's just a property of your system. So that could be something like a squid jump from an elected neuromag system. That didn't really happen in the real world, but unfortunately you measured it. And these are massive signals, much bigger than anything the brain's producing. Go back a second. Won't let me. Okay, so these artifacts. I'll just raise these artifacts. Often have a design of their own. They're not just randomly distributed throughout all of your conditions. It might be the case that sometimes there's more of an artifact in one condition than another. Think about a task where there's some surprise. You're looking at the surprise trials and you're comparing them against trials without surprise. Maybe people blink more in those trials through surprise. Now that blink is going to produce an artifact, as I'll kind of touch on later. And if you don't remove it, it doesn't matter how much averaging you do, you're going to average that blink into your data as well. So you're going to need to remove that blink. Anyway, just another nice thing about pre-processing is that after you've kind of gone through everything in SPM, you're going to end up with just a .dat and a .map file for your data set, maybe lots of images and all that kind of thing, but it will be something which will be interpretable by lots of people, especially if they've done this course. But at the start, and maybe what you've got right now is you've got a lot of files and you know what they all mean. But trust me, maybe in a year or 2 years, even you won't know. So it's quite nice that pre-processing also kind of organizes your data for you, I think. Which is quite nice. Okay, so what do I like to think? What have I learned about pre-processing? So it's generally advisable to treat all data the same. And what do I mean by that? Well, usually you'll be making a batch or running the same script, applying the same filters to all conditions, and in that sense you're treating all data the same, right? And that's important because, as I say, these artifacts in your data might be orders of magnitude larger than the signals. So if somehow you attenuate an artifact 80% in one condition and 90% in another, you'll very likely find a difference, but it's nothing to do with the signals you were trying to measure. I guess it's a bit more complicated than that, really, but that's the idea. So you might be thinking, well, as long as I just apply the same things to all my data sets, that'll be okay. But you might find that you've actually designed, say, your filter parameters based on one condition rather than another, and I'll come on to that on a later slide. There's actually often some manual steps when you're doing your pre-processing. So that might be visually inspecting your data and selecting bad trials. And then, if you're going to be doing that over a week, you might find that the principles that you're applying to select those bad segments can change. And you'll have to be really careful about that. So again, I'll come on to that a bit later. Anyway, treat all data the same. That's the aim anyway. It's also quite nice to check what each pre-processing step is actually doing. So this is quite nice in SPM. You can visualize your data in some way, or in MATLAB, or however you prefer, before and after each step. Apply a filter, or rather, plot your time series, in segments. Have a look at that. Be familiar with it. Apply a filter to your data. Plot it again. See if it makes sense. And that's nice, not just so you know what you've done, but also to check you haven't made a mistake. Maybe in the GUI you added a 0 where you shouldn't have, and you've got a 10 Hz, not a 1 Hz low pass filter, high pass filter. So that's a good idea generally. And it's also good to think about how different pre-processing steps interact, and I'll kind of highlight these as we go along. Sometimes these technically matter, but you can do it both ways, and you'll see it's not going to affect your results. But other times it really is important, and I'll try and highlight that as we go along. And really importantly, ensure that you're able to report what you've done to people, and also be willing to defend it, because you'll often find a reviewer will say, oh, you did that. If you've managed to report it properly, they'll say, oh, you've done that, I wouldn't have done that, why did you do it? So you should be, willing to justify each step that you take. And I put the part about making sure that it's replicable as well. That's particularly important for manual steps. You'll want to make sure that you're able to sufficiently describe what you did. Don't just say you visually inspected, or ideally don't just say you visually inspected bad trials. Try and say what you used to determine a bad trial, if you can, for example. Okay, so what steps are we going to cover in this talk? Well, I was thinking about how to do this, and I'm going to go a little further than just sort of saying what can be done in SPM and recommending parameters and all that kind of thing, because that's going to be covered in kind of the walkthroughs and the practical sessions. So instead I'm going to go through a few of them and go into some depth, and hopefully an appropriate amount of depth as to what's actually being done at each pre-processing step, and what you need to consider, and hopefully that'll go okay. But briefly, we're going to cover, I'm going to cover digital sampling and resampling, temporal filters, or filters usually, how to identify bad channels, epoching your data, re-referencing, specific to EEG, so sorry MEG people, and also then just the very broad topic of dealing with artifacts. But before I get to that, let's talk about what's happened to your data so far. So I kind of said that pre-processing is the series of signal processing steps that you take after you've recorded your data and before you do your analysis. But usually there's actually been some kind of pre-processing which has occurred before you've exported your data from your system. So I'm going to talk about these two a bit later. So sampling frequency, that's kind of predetermined unless you've not collected your data yet. But I'll go through all the considerations that you need to have for that on the next slide. Hardware filters, so essentially a kind of an electrical temporal filter. These will often have been applied, and again, I'll talk about them on a later slide. What I won't talk about though is electrode impedance, and you just kind of need to be aware of this. So when you record EEG, hopefully your system will record the impedance of the electrodes at the start and maybe at the end of a recording. And you should keep an eye on these, and you should note them down so that you can check that you've got them all under 5 kiloohms or maybe 10 kiloohms, but ideally less than 5. And why is that? Well, it's particularly important for low frequencies, as you can see in this nice figure from Kappenman and Luck. So what they've done there, you could ignore the stuff about temperature, but they've essentially recorded the same signals from the same people with high impedance and low impedance electrode setups. And you can see that for the low frequencies, that kind of effect on resistance that varies across frequency that impedance has. It's added noise essentially to the high impedance electrodes for those kind of sub 10 Hertz frequencies, and you can imagine how that might be a problem if that varied across your conditions, or even across sort of units of observation. It would be quite problematic. So make sure your impedance is low, and certainly make sure you record it. Also be aware, and I'll talk about this on the re-referencing slide a bit more, but be aware of what your online reference was. Usually it actually doesn't really matter too much, because you can re-reference, but you do need to know what it was. All this information should be, the sampling frequency and the online reference, all of that kind of thing should be stored in your files, and SPM will read it in, hopefully. Anyway, for MEG data, you should be aware that some kind of signal space separation, SSS or max filter or a Maxwell filter, depending on on what you've used will have been implemented. I'm not going to go into the details of this, but essentially all that does is it takes Maxwell's equations and spherical harmonics, and it uses those to model your data as two spaces, internal and external. And the idea is that if all goes well, the internal space will contain all of your signals of interest, and the external space will contain noise. And by separating them, you can obviously dismiss the noise. And you can see this has a drastic effect on your data. So in this nice paper from Jazz and colleagues, which compares a bunch of different pre-processing steps, well worth looking at, you can see how max filter or Maxwell filters with their different implementations, drastically affect your data. So be aware of those parameters, sort of what's happened to your data before it reaches SPM. Anyway, let's talk about digital sampling and resampling. So there's not a huge amount to say about this, I guess, but I'm going to try. Consider that the kind of the real signals in the world, they're analog, they're continuous in nature, but the way in which we record our data is pretty much always discrete. We take sort of little bite-sized pieces of that data in time and digitize it. And I say most because early electrophysiological recordings, not just EEG but like ECG and that kind of thing, they would have been done using some sort of electrometer, which would have actually scrawled a bit of paper, and that would be an analog recording. Anyway, everything you'll be doing in SPM will be, of course, digital, and hopefully it will have equidistant sampling. I'm only mentioning this because it allows us to talk about hertz. So the idea being that the space and time between each sample should be equivalent throughout your whole recording. You don't want to sample, for 20 milliseconds and then 21 milliseconds later you do another sample and then back to 20. It will always be uniform, hopefully. And when I say hertz, that just means samples per second, or the inverse of the time between samples. So 20 hertz, 20 samples per second. Anyway, so you might think, oh, maybe I'll just sample at the highest rate I can, and that's not a good idea, because the lower your sampling rate, the more efficient everything's going to be, not just in terms of disk space. We can end up with some pretty large files in Neuroimaging, but also in terms of how long it will take to run these pre-processing steps, perhaps some of your analyses, depending on what you're doing. It can really slow things down, and you'll spend a lot of time just looking at that kind of progress bar in SPM. So you want it to be as low as you can make it, really. And there's really only one rule, which everyone will say follow, which is Nyquist-Shannon. So the idea here is that you need to exceed the Nyquist rate, which means you need to double the highest frequency of interest that you'd like to observe without aliasing. What does that mean? Well, I've got this nice practical example for you on the right. So here, the sort of dark blue line on the left panels is an analog recording. It's actually just very highly sampled, and it's a 20 hertz sinusoid. We can just see a snippet of it here. I've also added some noise, and I've shown what happens if you try and sample that at 30 hertz, which would be below the Nyquist rate. And you can see that if you were to connect those red dots together, kind of in your mind, I've not done it. If you were to connect those red dots together, you would see that we don't actually have much, if any, information about the original 20 hertz signal. And indeed, if you look at the right at the power spectral density, which just shows the power per frequency band, power per frequency, you can see that it's kind of introduced this peak at 10 hertz, as well as actually managing to produce some information about the 20 hertz. and that's aliasing. It's some interaction between the sampling rate and the signal of interest, which is above the Nyquist rate. Yeah, so you can see why that might be an issue. And then if you were to sample at a higher rate, say 64 Hertz, which is now above the Nyquist rate, you will see that, yeah, it looks like we've got a nice kind of summary of that analog signal if you were to connect those green dots together. And on the right, you can see there's no phantom piece, there's nothing strange going on. It's pretty much a perfect, it's pretty much perfectly captured that peak at 20 Hertz, and it matches the blue, so that's good. And if you were to increase the sampling rate again to say 128 Hertz, you can see, yes, it's the same deal. It looks a little nicer in the time domain, but really no extra information has been captured. So I suppose in this case you might think, yeah, let's go with the green. What does this mean practically for actually doing EEG and MEG? Because if you think, oh well, the signal I'd like to observe, maybe I'll only be looking at alpha, 12 Hertz, or 10 Hertz being the central frequency. Oh, then I just need to sample at 20. Yeah, I don't actually know what would happen if you did that, but I guess all of those high frequency signals would alias. So then you might think, okay, I have to double the sampling frequency. I have to set my sample frequency at double the highest sort of noise that I've recorded. But by the time you've got 50 Hertz and it's harmonics and other high frequency signals, that'd be a very high rate. So to be honest, what I think most people do is they just go with 500, or they will say start at the original sampling rate 1000, and then they will plot the PSD and then downsample to 200, plot the PSD again, see if any aliasing has occurred. But yeah, to be honest, people talk about the Nyquist rate and then it seems to never be used. So yeah, with a pinch of salt, I guess. Oh yeah, there is another consideration. So it's actually not just the Nyquist rate. That's not the only parameter you need to think about. Sampling rate specifically. It's you should also think about the interaction between the number of samples that you have and the discrete Fourier transform, the DFT. So PSDs, that was kind of the PSDs I was showing on the previous slide. Those are based on extremely long time series, not just that 200 millisecond snippet which I showed. And that allows you to get a nice kind of high resolution discrete Fourier transform, which is what you can see on the right. It's got a nice sharp estimate of amplitude by frequency. But it's actually the case that yeah, there's sort of almost a one-to-one mapping of the number of samples in the time domain and the resolution of your Fourier transform, and you can see that here. So in this situation, we've got a couple of variables. So moving from left to right, I've taken the duration of a time series, so 100 milliseconds on the left, 400 milliseconds in the middle, and just over a second on the right. And moving from top to bottom, you've got a sampling rate, so 500 Hertz, 2000 Hertz, 8000 Hertz. And what you can see is that where I've put in a 180 Hertz sine wave signal, so I've got a high frequency signal, you can see that when you've got a short duration, even though we've exceeded the Nyquist rate by quite a bit with 500 Hertz, we can't see a peak there at all. And even as you increase the sampling rate to well above the Nyquist rate, 2000 Hertz in this case, you can see we can see a bit of a peak, but you wouldn't want to rely on it too much because some noise might exceed that. And really, it only becomes the case that once you have a lot more samples, so in this case 1.2 seconds and 8000 Hertz, that you can start to get a good estimate of that peak. So just think about that if it's going to be relevant to your work. Right, let's talk about temporal filters. So this is a massive subject. Temporal filters are everywhere, not just in neuroscience, of course. And why is that? What are they good for? So often we record things in the time domain, and you can think of what we record as the sum of many periodic signals. Essentially lots of sine waves, all summed together. Not in phase, they're phase in their amplitude. And yeah, it can look messy. If you want to do your analysis in the time domain, you won't necessarily be able to do it if you want to just look at one of those signals. But if you look in the spectral domain, so this PSD on the right, you can actually see that in this case where we've got a 10 Hz and a 50 Hz signal, you can see they're actually really nicely separated. That's the kind of thing you'd want to analyse, right? So how do we take this nice separation and apply it back to the temporal domain on the left? Well, we can apply temporal filters, which I'll talk about a bit more on the next slide. But generally speaking, you want to design a filter so that it preserves a passband. So a passband being a range of frequencies which you would like to keep all of that information exactly the same. And you want to attenuate the stop band. So the stop band being those frequencies which you'd like to kind of decimate. And that allows us to talk about high pass filters, which attenuate low frequencies, and low pass filters, which attenuate high frequencies. You've also got band pass filters, which kind of have two stop bands, so a stop band either side of a pass band, and you have band stop filters, which have two pass bands and one stop band. And yeah, you can see these are extremely powerful. So in the green, I've just made a high pass filter at 30 Hz. Kind of strange, you would probably never do this, but you can see that it has attenuated those lower frequencies in the spectral domain, and also you can see they're no longer present in the temporal domain, which is brilliant, isolated the signal perfectly. And likewise, if I were to flip that round, so in the black I have, again I think it's at 30 Hz, and instead it is now a low pass filter. And you can see that all that remains is that kind of low frequency sine wave. Brilliant. Okay, but how do they work? They're not just magic. I think fortunately, in the simplest case, they're actually quite easy to understand. Temporal filters just use a moving weighted sum of samples, which is a type of convolution, and that transforms some input signal to produce an output. And in these lovely figures from de Chavannes and Nelken, which is another paper which you should definitely read if you're interested in temporal filters, they kind of take you through exactly how they work, how to report them, all that kind of thing in a lot more detail than I can here. Anyway, in this filter, in this figure, you can see that because it's a moving weighted sum or average, the many samples from the input affect a single sample in the output. So that's kind of what you can see in the top. And another way of phrasing that is, for every sample in the input, there are many output samples which are determined by it. And that necessarily means that filters are going to sort of smooth your data or smear your data through time. They drastically change what you've recorded. And just to say that these weights are essentially the filter design, and they're normally called the impulse response. And briefly, they're called the impulse response because if you were to set your input to be all zeros, 1, 1, an impulse, and then all zeros again, by applying a filter to that impulse, about that impulse, you would then see the weights in the output. Anyway, so as I say, the signal is now smeared across time, and that means that the exact type of filter and the way it's done can affect the timings of, say, the peak of an ERP. So in this nice figure from Akunzo and colleagues, you can see that in the original data, in the red, the peak is actually sort of slightly ahead in time. Is that right? thought it'd be the other way around. Anyway, and in the black, you can see the peak is actually shifted after applying what's called a causal filter. So a causal filter can only apply weights to samples which have occurred before it in time. That's kind of a take on electrical filters, which kind of happen in real time, and they can, of course, only consider previous samples, I suppose. The other thing you can do is you apply a filter forwards if you've already recorded it, and then backwards, or essentially have weights either side of the output. And what this means is, the peak isn't going to shift around, but something kind of weird happens in that you've got a time point in the present now affecting samples that occurred before it. So again, you should think about how that's going to affect your analysis. If perhaps you're trying to decode something and find out exactly when it happens, think about whether essentially your use of a filter has meant that decoder now has information from the future at a current sample. All sorts of strange things can happen. Anyway, how are filters implemented in SPM? Well, this is where that example breaks down a bit, to be honest, but the principles do remain. So SPM uses Butterworth filters, which were originally electrical filters designed by Butterworth in the 30s, and they're optimized to preserve the passband and attenuate the stop bands to kind of a maximal extent. And they try and minimize what's called the transition band, which is that sort of gradient between the passband and the stop band. So you don't really need to know exactly how they work, but I guess be aware that they have an infinite impulse response. So like I say, that nice simple example I had with those weightings, it kind of breaks down a bit because that weighting is now of infinite length. Although in reality it does tend towards zero. So SPM implements these filters for everything by default, so for bandpass, bandstop, high pass and low pass filters. Essentially it uses filtfilt in MATLAB, which is an acausal or bidirectional filter. So it's kind of that second example from the Akunzo paper. And the default order of the filter is 5 SPM. And what do I mean by order? Well, again, I'm not going to go into it because it's not really my world, to be honest. But the filter order determines the gradient of that transition band. So a filter of order 2 or a filter of order 4 has twice as steep a gradient as a filter of order 2. And so you might think, why don't I just always apply, why don't we do a filter of order 10? And the reason for that is computationally it becomes quite intense, and also the stability of the filter breaks down. And briefly, that's just because there's some feedback, ongoing feedback that occurs when designing a Butterworth filter between the data and the filter parameters. That gets increasingly complicated as you increase the order. So anyway, practically, I would just leave everything on the defaults and just try and filter out, anything that isn't your signal. And what are the considerations to have when using these temporal filters? Because, like I say, they drastically change your data. It's well worth looking at your data first, applying a filter, and then looking at it again, and you'll see it looks like a totally different data set. And that's because essentially it is. This is actually something I mentioned, I think, earlier, where if you design a filter for just one of your conditions and then apply it to all of them, you may find that you're going to affect your results. So again, in this example from Akunzo, they show that depending on whether you apply a high pass filter at 1 Hz or not, and they had loads of examples, you either find a difference in the ERP in the happy condition in this case versus fearful and neutral, or you don't. And that's because there's some low frequency signal in the happy condition which isn't present in the others. So I think this is quite an extreme example, and hopefully normally you've got kind of the same signal properties that are being modulated in some way across your conditions. But if not, be aware of what might happen. And I think the best thing to do is just look at what other people have done in your field, and this is why it's nice when people report all of these things fully. And yeah, always make sure that you report everything fully so that people can be aware of what you've done, because it might be that these pre-processing steps are actually incredibly important. Yes, I think I'll leave it there. Okay, so it's very important to identify bad channels in your data. What I mean by bad, well, in EEG, maybe a cable, a lead was moving and you've got some cable artifacts. I think if you attend some of the OPM MEG talks and demos in this course, you might hear about some of the system-specific issues that we've had there regarding bad channels. Generally speaking, it's very obvious. There's just one of your channels is standing out, like you can see here in the red, as having some sporadic noise, or perhaps if you were to look at the power spectral density of all your channels, you might see that a couple of them are significantly higher than others, and there's just something wrong with those channels. Often, actually, it's the case that you're just sort of given a list. So you use your local SQUID MEG system, and it might be that you're given a list of channels which are very likely to be bad and which you should just remove. Anyway, there's kind of a balance to be struck here between removing channels. You have them for a reason. You have them to get good spatial sampling, high dimensionality in your data. You don't just want to be removing them. So there's a balance between keeping trials in and removing more time points. And generally speaking, if you remove a time point, you remove that time point from all channels. All you could remove essentially this segment. I'm not sure you can see my mouse. You essentially remove a whole trial rather than just one channel. And there's a balance there, and there's no right answer, I'm afraid. You just have to go in with some principled approach. Either remove all bad channels, or all bad trials, or something in the middle. But whatever you do it early. This is where it really interacts with other pre-processing steps. So especially where you've got a spatial filter, something like max filter, you don't want bad channels in there because they're going to sort of project across many other channels, and then you'll have lots of bad channels, I guess, and you might not even be able to identify them. So that would be a problem. And you should also think about when the order of applying filters before you start to identify bad channels. So in SPM, there's a few automated methods, and these might say look for squid jumps. So that'd be quite easy. A squid jump is a kind of a rapid increase from one sample to the next. So you could just look at, diff in MATLAB, and that would find kind of a quick rise, and you could say that's when the squid jump occurred. So now remove 50 milliseconds either side of that, something like that. However, if you've applied some filters, some temporal filters to your data, you might find that transition is no longer so sharp, and these automated methods, they might struggle to identify that squid jump. The same applies to eye blinks. Whether you're doing that visually or automated, filters are going to really drastically affect what those eye blinks look like. So just be aware of that. And then, epoching data. So this kind of comes into averaging and various other things, but it's pretty much the most powerful thing you can do, I guess. So I mentioned earlier that you're going to have some knowledge of what's happened when in your data. So some events table, which will be when a stimulus was presented or when a person responded. And hopefully you've got lots of these. And in order to average across them, we do some epoching. And epoching is not really a word. It only seems to exist in EEG and MEG. I guess it's making epochs. And usually you define a start point, and then you can sometimes go back a bit, often a baseline period or just some kind of pan event time. And then you go forward by sunset period. And SPM kind of likes these to all be of the same length, by the way. So think about that when designing your experiments. And yeah, there's not too much to say about it, other than, of course, this allows you to do some stats. Could be as simple as just averaging across epochs within two conditions, doing a t-test between them, or maybe you want to preserve all of the epochs and they all kind of go into a GLM. But you'll hear all about that in some of the other talks. I guess the only thing you should be aware is that often when you chop up your data like this, there's some baseline correction that goes on. That could be based on the mean of a baseline period, say the second before an event. It'll take the mean of all those samples and subtract it from the whole data, or it could be the mean of all samples in the epoch. So just be aware that I guess if you were to stitch them all back together, they'd be offset in some way. Yeah. Right. So let's talk about. This is a bit of a change in pace, but let's talk about referencing and re-referencing of EEG. So, practically speaking, this is all very easily handled in SPM. So you should know what the online reference was, and I'm going to talk about what that means in a moment, but you should know what it is, and you should decide whether you're going to re-reference to some other scheme or montage. So, generally speaking, if you're going to do some kind of source analysis, you will want to use the common reference or the common average reference, which takes the voltage from all channels and which will have a mean of 0 and uses that as the reference to each of the individual channels, and from that you kind of get an absolute measurement. Often for just doing a sensor level analysis, there's a lot more approaches, but generally speaking, people like to use the average of kind of a right and a left reference, which would often be M1 and M2, the mastoids, or it could be earlobes, something which isn't going to have too much neural or muscle activity, and which isn't kind of spatially biased, I guess. That's why it's the average of the two sides. Whereas you'll often find that the online reference is, say, CPZ right in the middle. However, for central level analysis, you can also take a common average, and that's what I normally do personally. Right, but what does all this mean? So this hopefully was covered in one of the earlier talks about the origins of these signals, but just to recap, EEG is essentially measuring, at each electrode, you're measuring a relative potential between two points, two electrodes. So if it's online, it was CPZ, something like that, and T7, it's the relative potential between these 2 points. But not really. That's actually not the case, because there's also the ground. And this really is kind of getting into the weeds, and it does not matter. But I think it's kind of interesting and something worth being aware of. So when amplifying a referential signal, like what I've just described, one side of that, I don't know the details of electronics to be honest with you, but one side of that is kind of bound to the common ground in the amplifier, and that will contain a lot of electrical noise. All these circuits just contain a lot of noise, which you do not want in your data. So essentially you've got, if you were to just do your active to your reference, the reference is going to contain a lot of noise. So when you do active minus reference, you're going to be left with the difference between the two in terms of what's going on at the scalp, plus negative noise, so essentially just plus noise, which is a problem. So how do you get around that? So I think they're called differential amplifiers. So these will take two referential signals. So instead of doing the active with reference to the reference, you do the active with reference to a ground, and we call it the ground. It's kind of a virtual ground, a little bit odd. Anyway, that's just another position on the scalp. And that means that now all of that noise is in the ground. The ground is being treated as the reference. And then you also do the reference versus the ground. So in this case, the ground is the reference, bear with me. And again, in this case, the ground has all of that electrical noise. So at this point, you've got A minus G and R minus G. So in order to recover A minus R, you do A minus G minus R minus G, leaving you with A minus R. Yeah. I've explained it now. You probably never need to think about it again. I think it's kind of cool though. Yeah. So you should be aware. I've already talked about this. Yeah. So you can use the common average, which you can just do very simply in SPM by selecting all of them. Or for center level, you might do M1 plus M2, or the common average. If you're doing some clinical work, maybe, which is very unlikely in a research setting, but say you are, just be aware that you can specify some quite interesting montages. This one's called the double banana, which you can see on the right. So this would allow you to look at things like a phase reversal. So they sort of reference from, in this case, FP2 to F4, F4 to C4, C4 to P4. And you can imagine how if there was some signal in the middle here, that the phase of that signal will switch as you move along. But like I say, not really used in a research setting, just kind of interesting. Right, so this is back. MEG people can wake up. Because this affects everybody. So how do we actually deal with artifacts specifically? I talked about filters, and that will actually help deal with artifacts. I talked about epoching. That might help a little bit. But yeah, sometimes, as I mentioned earlier, artifacts can have their own design which gets around all of these things. Perhaps they overlap in time. Sometimes maybe more in one condition than another. Maybe sometimes you can't filter them out, like an eye blink which occurs at low frequencies, and maybe you're looking at theta. You can't apply a temporal filter there to remove the eye blink. So what can you do? There isn't a right way to do this. You can either try and remove all contaminated segments, so all sort of time points or trials which contain an artifact, and then you might lose, I don't know, half your trials, or maybe you won't lose many at all. It really entirely depends on your paradigm. I say if you're not going to lose many at all, do this. If you're going to lose half, try something else. So that something else is trying to correct or remove artifacts from your time series without removing whole time points. And to do that, you usually use some spatial information, which I'll talk about. And the other thing you can do is you can just ignore the issue. Like I say, that will probably only work if you've got really clean data, or if they don't have any sort of design of their own. And then you can just use the power of averaging across all of those epochs, and that really will help a lot. Yeah, so here's just a nice example I borrowed from somebody else's slides, but you can see here how different artifacts kind of appear in your time series. This kind of requires some expertise, which I'll talk about on the next slide, but you might have some continuous muscle activity, which is this kind of high frequency signal you can see, or a transient sort of, that almost looks like the jaw to me, sort of burst of muscle activity. An eye blink, that looks like a filtered eye blink to me, got quite a low frequency, kind of half a second duration artifact. And then you've got your ongoing signal, something like alpha, which is often visible in the time series. Yeah, so you can see it might be a bit of an issue. So, in the simplest case, just rejecting, say, all trials that contain artifacts. To do that, you first need to identify all those time periods, which is not necessarily simple. So one way, you can kind of look at that time series on the previous slide, and you can imagine you might be able to just select time periods that contain an eye blink or muscle activity. And yeah, you could then just say, if a trial contains any of those, remove the trial. Fine. And yeah, that's kind of what I like to do, I think. Usually when you've spent so long designing an experiment, so long collecting that data, you're going to be spending a long time analysing it. spending a week or something like that, or depending on how much data you have, just scrolling through and labeling bad segments is well worth doing. Yeah. There are automated methods as well, certainly plenty in SPM to look for things like eye blinks, and again just consider how earlier pre-processing steps like filtering, etc. Influence that. There's also kind of semi-automated methods. So this is actually from Field Trip, but it's in the SPM manual. It's highly compatible with SPM. You can use this reject to visual function in Field Trip, which allows you to create a summary statistic across trials and across channels, and it allows you to then just sort of drag and select anything which looks a bit out of the ordinary in a semi-principled way. So in this sense you could say, oh, I plotted the kurtosis of all trials and I removed trials with a kurtosis of greater than X. Yeah. So you can do that, and then you can use that to just reject any data. Fine. But what do you do if doing that would be impractical? So you've got some one-shot learning paradigm or something like this, and you don't want to remove too many trials. Well, in SPM, you can do a few different things. One is to use SSP, signal space projection. So I think this is going to be covered in the demo, but briefly, you use some automated or manual procedure to select artifacts, and rather than removing them, you treat them as an event. You sort of get some pan artifactual set of trials almost, and you use these to build a couple of subspaces orthogonal to each other, and then you remove the one which contains the artifactual data. Yeah, and that works really well. It's very efficient. Another common approach is to use ICA, but to do that you'll have to use some external tools. So in Field Trip it's often used for removing artifacts, and there's actually automated methods, particularly for EEG, so IC label, implemented in EEG Lab, and that's not only used for rejecting artifacts, it's actually also used for kind of isolating and collating neural activity as well, which is an interesting approach. And just consider how these methods affect the rank of your data, because you're essentially removing some piece of information across all channels, so you're reducing the dimensionality of your data, and you just need to be aware that if you remove loads and loads of components, you might need to change some later parameters when doing your source analysis. But yeah, generally speaking, these are powerful tools. ICA is probably the most common method, to be honest. Yeah, but not in SPM. Yeah, okay, so I think I'll just leave it there. I'll be around hopefully to answer any questions that you have, and yeah, thank you for listening. Hi everyone, I'm Nick Alexander. A postdoc here